thing that you may have noticed different between our two models and our two lectures. But first, Jason Wolf has arrived, who will be the third person speaking to you. But anyway, you may have noticed that I put up a set of 8 different partitionings for the 9 genotypes, which I identified as additive and dominance and additive by dominance, et cetera, et cetera. If you look at those, you'll see that they're different from the ones that Dr. Chevroud put up, that his numbers are different than mine. And you might ask why that is. Well, it turns out that when you have 9 genotypes, you can fill them in any way you want to, and you can make them orthogonal, independent of each other, but only for one set of circumstances. And so what I did was I chose a gene frequency of 0.5, and I've got a set of 8 different interactions that are independent when the gene frequencies are 0.5. Dr. Chevroud, on the other hand, following in Falconer's model, said let's start with a situation where you don't have any gene frequencies and just look at the deviation from the average of the 4 double homozygotes. I think that's right, isn't it? Or is it 9 genotypes? I'm not sure. But anyway, his are independent in this space where gene frequencies are not included. And so that's why the 2 are different. Essentially, you can choose your 9 genotypes and choose your circumstances, and you can find your own 9 independent contrasts that are of interest to you. And what was the...
Well, I think I gave equations that related mine to yours.
Yes, they can be related in between. And there's actually a paper out there, and that's the...
Alvarez, Castro, and Karlborg, I think, in genetics in 2006 or 7.
It's something right around there. I don't remember.
That should be enough.
But anyway, they give a general formula that allows you to translate between these different forms very easily. Okay.
All right. Well, we weren't on camera yet. Now go. Okay. We're being recorded, so watch what you say. So now I was going to talk about pleiotropy, which was the last part of the genetic architecture of the list of questions. And pleiotropy, the way I think of pleiotropy, which is the effects of a gene on multiple different traits. So one gene, many traits. Polygeny is many genes, one trait. Pleiotropy, many traits, one gene. And this was, my view of this is very much colored by a book called Morphological Integration by Olson and Miller. It was published in 1958, I believe, by the University of Chicago Press. And it was reissued later as a paperback by the University of Chicago Press, quite a bit later. This must have been in the mid-1980s. Olson and Miller's idea of morphological integration had a pretty big impact right away. But as you see here is a graph of the citations to the book by year. The first 2 on this axis are the 1960s and the 1970s. So those are kind of composite. I need to put my glasses on so I can see. Okay, yeah. The 1960s and 1970s here, and then 1980 on. And you can see that there's a dribble of stuff all along, and then for some reason, around 1998, 99, it got popular. So this tracks the popularity of the book and through time up to 2008, which was the 50th anniversary of the publication. So unfortunately, Olson and Miller both passed away, but their book became really important 50 years after they published it. So the idea of morphological integration is concerned with the integrated relationship among characters. So just simply the the integrative relationship among morphological characters. And there are several different forms of morphological integration that are related to one another. The first one is functional and developmental integration. And this is interaction of morphological elements in the performance of some function or developmental process. So an example on function would be the length of the upper and lower jaws. Teeth need to interdigitate properly, and teeth will move around in your jaw in order to interdigitate correctly. But if the difference in jaw length is too large, you end up paying thousands of AI to the orthodontist to get your teeth to interlock properly. Now, genetic integration refers to morphological elements that are inherited together due to pleiotropy or linkage disequilibrium. These are represented in the genetic variance covariance matrix of quantitative genetics. The importance of linkage disequilibrium in genetic integration is highly variable. It depends a lot on the structure of the genome as to whether most genes are in linkage disequilibrium with other genes because they're near each other on chromosomes, only a few chromosomes, versus species that have many chromosomes and largely independent inheritance of the individual genes. We have evolutionary integration, and this is that morphological elements that evolve together. So function together, are inherited together, evolve together. Functional integration leads to genetic integration, that in turn leads to evolutionary integration. This is the basic idea that Olson and Miller had. I gotta use a littler finger to step up. And with integration, subsets of integrated characters evolve to form semi-independent modules of function, inheritance, and evolution. And that these modules of function lead to the modules of inheritance that leads to the modular expectation for evolution. So they mimic one another. Now, this is an idea that Rupert Riedel had, if you ever read the book Order in Living Organisms. Now, Olson and Miller didn't know quantitative genetics, even though it had been around for, 1950. It had been around for 40 years or so. They didn't really know about it. This is before computers came about, so their methods were different too. They invented their own forms of cluster analysis to try to find groups of related traits. So reading it, I tried to translate that into genetic or quantitative genetic framework. And this was done with animals from a skeletal collection of the population of rhesus monkeys on Cayo Santiago off the southeast coast of Puerto Rico. Now that might be a strange place to find rhesus monkeys. You might not think of it as their native habitat. They do very well there. They've escaped off, not of Cayo Santiago, this one's too far out. They've escaped from other islands off the coast and come inland and are raiding farms, and now they've started shooting because they're a pest. And you can read about the rhesus macaque pest problem in the cane fields and cornfields of Puerto Rico. The animals happen to be there because one of the founders of behavioral primatology, Clarence Ray Carpenter, went and bought a bunch of rhesus monkeys in Calcutta, brought them back to the United States by ship, and got to New York, sold half of the animals he had collected for biomedical research, thereby paying the fare for the monkeys, and bringing the rest of them down to this small island. It's 40 acres. I don't know how many hectares that is, but it's a small island. With two major portions here connected by a small isthmus. And he let them loose. And he actually brought some gibbons too and some other kinds of primates. But then that was done just before World War II. During World War II, things changed. The Nazis had submarines coming into the Caribbean, and the U.S. Navy kind of took over everything and didn't allow the researchers out there for fear of spies or people getting hurt or whatever. Until very recently, there was one of these islands that the Navy used, U.S. Navy used for target practice. It didn't have a lot of vegetation left on it because they were pretty good at hitting the island. So the island is provisioned. They bring out monkey chow and put it in hog feeders. And the animals lift the doors open with their one hand and stuff the food in with the other. And there are rainwater catchments that catch the rainwater for them to drink. Being provisioned like that means the population grew at an incredibly high rate. And animals have had to be removed from the island from time to time. And some of those animals have ended up in the skeletal collection of animals from Cayo Santiago. Other animals, most of them, found their way in there through dying on the island, and the behavioral researcher coming in and picking them up, rendering them with, putting them in a water bath out on the island for a few days. All the flesh is gone, and you just have the bone. It's very effective out there. In 1955, Stuart Altman, another dean of primatology, he came and he tattooed every animal. And since that time, they've kept track of the maternal genealogy from 1955 until today. When I studied these, there were 500 skeletons in the collection. There are now 3,000 animals in the collection in these pedigrees. For the early part, we don't know who the fathers were. More recent times, paternity testing's been done with microsatellite markers for rhesus monkeys. So in more recent times, you can identify both the mother and the father. We did have samples from the animals back from 1968, and they were kept in a freezer in the lab I shared with Donald Said, who was a behavioral researcher that ran the island in the 60s and 70s. A painter decided he had to paint where the freezer was, so he rolled it out and plugged it back in to a dead outlet. And all the samples went bad. I haven't been backed up since 2001. So that's why I still have that picture from 2000. To back up, I'd have to show up, plug into the university network at the right time. So this is a rhesus macaque, a mother and her infant. This is we kept track of natural lines. This picture is a series of 4 sisters. So females stay together and are the core of the social groups. Males, when they reach maturity, migrate out to another social group. And they'll migrate around during their lives. But it's the female core that's kept track of and that we use. I'm very proud of this picture. This is a drawing of a rhesus macaque, and I colored it in myself. And I stayed within the lines. Sister Maria Nia would be so happy with me. Because that was a big problem when I had in first grade, staying inside the lines. I learned to outline things first, and then color inside. That way my random movements wouldn't have too much of an effect. But broke the skull up into several different developmental units here, but they're also functional units. The oral unit here, the maxilla and the tooth row, the mandible. The zygoma here on either side, the nasal region, nasal capsule, orbital region. And then we have on the cranial vault, the frontal, parietal, and occipital. We also have the cranial base that you can't see on here. And copying Olson and Miller as best I could, I used the cluster analysis, Ward's hierarchical cluster analysis, with replacement, which is a special way of doing it. And then looked at the clusters I got and how they agreed with the measurements that fell into the same colors here. So each one of these colors would represent a region of the skull, and I would see if measurements in this region of the skull clustered together or not. And this is a measure of agreement between the theoretical clusters, which were called F-sets by Olson and Miller, and the observed clusters. And you can see that the agreement for phenotypic correlations, environmental, genetic, are all highly significant. However, when you have those 6 categories, 91.7 are correctly classified in the phenotypic, and a low of 58.4 for the genetic. The genetic is lower than the phenotypic and the environmental, in part because the average heritability is only 30%. So you have the best information phenotypically. Your genetic information is going to have a lot of error in it, and that error is represented in the lower agreement. But when we just consider the neurocranium or just consider the face separately, again, you had a fairly good agreement between the theory and the clusters of traits. So what this would say is that developmentally related traits are inherited together. That's what the finding is. You can also look at the correlations between traits that are within a functional set, within a module, versus ones that are between modules. And the R squared is given over here. You can see that the within module R squareds are many times higher than the observed, except for the genetic, again, with the extra error, still increases, but not by as much. It's a higher integration within a module than between modules, but not as strong as the phenotypical. The effective population size. This is a multivariate study. There were 56 traits. The effective population size, the number of individuals that I had secure breeding values for. The number of breeding values I knew was like 10 for this study. That's why there's so much error in the genetic matrix. Though there's 500 phenotypic values, there are only 10 breeding values. Then we moved over to a much nicer monkey. This is a tamarin from Brazil, right? This is an emperor tamarin. It's the cover of Hershkowitz's book. We went to the zoo yesterday and we saw lots of different kinds of tamarins. It was great. Incredible. We did a study again on 2 species of tamarins. The saddleback tamarin, Ciguinus fusicolus, and the cotton-top tamarin, Ciguinus oedipus. Now, the fashion these days is to make the 14 subspecies of Ciguinus fusicolus each their own species. This population was primarily Ciguinus fusicolus illigeri, but it also included hybrids with illigeri and 3 other subspecies, Leucogenes, Ligonotus, and one other. I can't recall what the other one is. And these are the cotton-top tamarins. And there were like 200 of these animals and 350 of these animals. They live in, well, their habitat is shown here. In some ways, you almost might think of this as a natural habitat for cotton-top tamarins, as there were hundreds in this colony. In Colombia, in the forests, we don't know how many there are. It was estimated, though, that there were 5,000 in captivity throughout the world, and probably about 5,000 in Colombia out in the forest. Nobody gets to check them out in the forest anymore because some entrepreneurs moved in, and they have a factory there that processes raw ingredients for export, and these guys have machine guns, and they don't like people coming around. So there's only a small remnant forest, but you can't study them. But in some ways, it's doing a great deal for conservation, because nobody's cutting down that forest either. I mean, you can see the cotton-top tamarins here in their natural habitat at Oak Ridge, Tennessee, which is a nuclear research station. These animals are there because top tamarins have spontaneous colon cancer. The fusicolis were there because the tamarins usually give birth to twins, and the twins share a placenta. The placentas fuse, so they share blood cells. So immunologists like to study the difference between self-rejection, rejection of your co-twin tissue, rejection of unrelated tissue from an unrelated, and then from another subspecies. So that's why they were there. Top tamarins can no longer be used for any terminal research because they're protected under CITES. So the population in captivity has dropped from 5,000 to about 1,000. So it's kind of a counterintuitive thing that happened. It all makes sense in a way. They have to be protected. But it's resulted in a lot less of them being around because they were useful for, they get Epstein-Barr virus, they get the colon cancer. There's a lot of things that are good models for human health. Here's an example of the genealogy. We do know all the parents, father and mother, because they were pair housed. And pedigrees, as you see, this one has 1, 2, 3, 4, 5 generations in it. Here are the skulls of the saddleback and top tamarin. This is a normal tamarin skull. It's got a low brow. The front of the face is flat. Cotton-top tamarins have this big lump up here, a greatly increased anterior temporalis muscle is responsible, or fits onto this big dome. You can also see that their face is less flat, the incisors sticking out some, and the length of the tooth row a bit longer. And we took three-dimensional coordinates, all of these locations, and we studied the same thing we did with the rhesus monkeys, divided the skull up into parts. And here again, this shows the average correlation between intramodular and extramodular traits. And you can see again that there's an enhancement in the modules. So again, developmentally related traits tend to be inherited together. Now, this kind of modularity can come from two basic genetic sources. I'm only going to be concerned with pleiotropy here. I'm not going to talk about linkage disequilibrium. But you can have modules by having individual genes that have opposite effects on different modules. If it's making the face module big, it makes the brain case module small, and vice versa. You could have that kind of thing, and that would produce the modules. So everything would show pleiotropic, all the genes would have pleiotropic effects across all the traits, but it would be a combination of positive and negative effects that would cancel each other out in some circumstances, making a module. Alternatively, there could be modular pleiotropy where genes only affect a subset of traits. Those are intended to be shown here. On your left is modular pleiotropy. You can see here are the loci. You have 8 loci listed. These are the phenotypes. So these 4 loci contribute variation to all 4 of these phenotypes, resulting in them being highly intercorrelated. And here we have a second set, a different module of loci and phenotypes that again have each locus affecting every one of the traits. And here we'd have a set of highly correlated phenotypes. But there's nothing from the loci up here going down there or vice versa. So the correlation between the two modules is 0. With antagonistic pleiotropy, here you really got to use your eyes. You can see there's a solid line here. You can see that there's dotted lines coming out from these guys. So each locus affects all 8 traits. It has a positive, so L1 has a positive effect on P14 and a negative effect on P5 to 8. L2, again, positive for 1 to 4, negative for 5 to 8. L7 down here, positive for 5 to 8, negative for 1 to 4. When you put all that together, you get high correlation within the set. The first 4 phenotypes and the second 4 phenotypes. But between them, the positive and negative effects cancel each other out and you end up with a correlation of 0. So we went to try to see what the results were, what kind of pleiotropy was shown in these mice. And a lot of this was done morphologically. These are measurements taken on the skull. And again, we have the 2 modules we can talk about broadly here, less detailed than in the primates. We have measurements on the face and measurements on the cranial wall. When we did our QTL mapping, we found 26 QTLs affecting skull morphology. 8 of them affected only the face and didn't affect the brain case at all. Another 8 affected the cranial wall, but not the face. 8 of them affected both the cranial vault and the face, but in the same direction. If they made the face bigger, it made the cranial vault bigger, and vice versa. So there were only 2 QTLs that displayed an antagonistic pleiotropy, opposite effects for the cranial vault and face. So clearly, modular pleiotropy dominates here. We also look at the same hypothesis in terms of the mandible, and each one of the processes of the mandible is developmentally and functionally independent of each other. The coronoid process is for the insertion of the temporalis muscle. If you cut the temporalis muscle in a rodent, which has been done, you find that this process resorbs. It disappears. Even if it's there to start with, it will disappear without the muscle pulling on it. The medial, the lateral pterygoid attaches to the condylar process, and then the angular process has an attachment from the masseter on the lateral side, you see here, and from the medial pterygoid muscle on the medial side. So different muscles, different bony condensations for each one of these 3 processes. This region here has the, and as we call it the masseteric region, but likewise the inside has the medial pterygoid. And it also carries the root of the incisor, which runs from here all the way up. We then have the body of the mandible here, which is mostly the alveolus for the incisor, and here the molar alveolus holding the molar teeth. Again, if you take the teeth out, the jaw resorbs, the bone disappears. So this bone is entirely dependent on related tissues. Without the related tissues, they wouldn't develop, or even if they have developed, they would disintegrate. Here's a picture of the measurements we used on a scanned mandible. You can see it's kind of translucent almost. It's kind of pretty, if you're a morphologist. And just like for the skull, we found here 27 QTLs affecting mandibular morphology. 20% of them affected the whole mandible, made the whole mandible either bigger or littler. 40% of them affected the tooth-bearing regions, but didn't affect the muscle attachments at the back of the jaw. And another 40% affected the muscle attachments at the back of the jaw, but didn't affect the alveolar portions. So again, we do have some general pleiotropy over the whole object, and we have modular pleiotropy for individual regions. Here we didn't get anything that caused the tooth row to be big and the muscle area to be small, or vice versa. This just shows where they are on the chromosome plots. You can see there's a lot of them and they're all over. This is traditional in QTL studies to show these kinds of slides, just to show that you did a map on the chromosome. Here's a couple of examples, and what I have here on your left is the additive genotypic values, and on the right of the chromosome, it's the dominance genotypic values. So here you can see this affects this muscle attachment region, that masseteric region, without really affecting the alveolar region at all. So this is a muscle attachment modular QTL on chromosome 2. You'll see that the dominance is just the same as the additive for this locus. The same traits that are being affected. The second locus here on chromosome 2 only affects the coronoid process. When the temporalis muscle attaches, it doesn't have effects anywhere else. So this is affecting one of those submodules, the coronoid module, without affecting even the other muscle attachment regions. But here you'll see that the dominance is different. Here's another muscular region one with a similar additive and dominance. Here's one where the additive and dominance are both alveolar, but this one's mostly on the incisor, while the dominance is mostly affecting the molar region. So again, differences between additive and dominance effects, but the effects are in the same module. So what's the pattern of single gene pleiotropic effects? Pleiotropy is largely modular, with single QTLs having exclusive effects on functionally, developmentally, or physiologically related traits. Now, once you put together dominance and pleiotropy, you get kind of an interesting phenomenon. I quoted from Lewontin last time, the 1974 book, and in that book, the major point of discussion is how is natural variation, genetic variation, maintained? It was obvious when you go into natural populations that they show quantitative genetic variation for traits, as of course agricultural populations did as well. And people were puzzled because in a Fisherian world where everything is additive, you shouldn't have much variation. The better allele should be fixed. So the only variation you'd have was new mutations arising, most of which would be bad and eliminated, a few of which would be favored and increased in frequency. But that would lead to a low level of genetic variation, very much at odds of what was seen. The solution to that has been to say that you see all this molecular genetic variation because it's neutral, that it does not have any function. The variations do not cause changes in the phenotype, so that they are not selected by adaptations involving those phenotypes. As a general explanation, that's explained. A lot of molecular variation is explained that way, but most of that I think comes from our ignorance. Clearly from the quantitative genetic, genes affect skull morphology, jaw morphology, and we know that there's been massive diversity of adaptation in size and shape in the jaws and in the skull across mammals that's seen as functionally important and adaptive. So there must be underlying variation underlying that, those kinds of features too, that haven't been detected by the molecular studies, perhaps because we are largely ignorant. And as we do our QTL studies now and we compare the sequences of the strains, we kind of know what to do when it's a non-synonymous SNP that changes an amino acid. When we're out in regulatory areas of the genome, we have no clue what we're looking at. You see a lot of variations there. Those variations are usually treated as neutral. I think that's more our ignorance than any information. Furthermore, if you're using molecules for phylogeny, you're assuming neutrality in the model you use to build the tree. So that made it convenient for people to take all this molecular variation and say it was neutral, because then you could build your phylogeny from it. So it turns out that when you have multiple traits affected by a locus, pleiotropy, and the dominance is different from one trait to the next, that this differential dominance can result in balancing selection on traits that are affected by our QTLs. People have struggled for the first part of the 20th century on to the 70s, trying to find balancing selection. And there's a few wonderful examples of it, but only a few. And so in 1974, people might have said balancing selection was responsible for the standing variation. and molecular studies weren't finding it. So we go back again to our number line, where we have our three genotypes, the two homozygotes, the heterozygote, the A and the D. And there's also a dominance ratio, which is D divided by the absolute value of A, which is just how big D is compared to A. And if you have two different traits, and they have different dominance patterns, an interesting phenomenon can occur. So here, capital A is dominant, small a for trait X. For trait Y, they're co-dominant. The heterozygote's right in between. So we take these two number lines and we make them into an X and Y axis. And we plot the two homozygotes on the XY graph now. And the way I plotted this, this is the typical kind of thing we see, is that all the traits are made bigger by the same allele. Rarely would we find that antagonistic pleiotropy that would require that would have a line pointing negatively. And this is called now an A vector because it's got two numbers in it, not just one. And then we can plot the heterozygote in its position. And the example I gave with the number lines before is shown here. So the average value of the heterozygotes, X is here, it's the same as for the AA homozygote. And Y is here, it's midway between the two homozygotes on the Y axis. So that's the dominance vector, because we have an X and a Y in it now. And there's going to be an angle between these two vectors. If this angle is 0, if the dominance of the two traits were the same, the heterozygote would lie along the A vector. It might be up beyond the homozygote or below the homozygotes or between them, but it would lie on that line. In that case, we don't get any leverage for balancing selection at all. But as that angle increases, we have stronger possibilities for overdominance of the heterozygote. So with differential dominance, there will always be a dimension, some linear combination of the original variables, that will be overdominant, even though no single trait shows overdominance. People have often looked for balancing selection and overdominance one trait at a time. And we have some of that for our traits, but not a lot of it. We can define something called the overdominance vector, which is the projection of the heterozygote onto the additive vector. So this is a 90-degree angle for the red vector. This measures the amount of overdominance represented by the two-trait system. And the length of the overdominance vector is equal to the length of the dominance vector, this one here, times the sine of the angle between the additive and dominance vectors. So I had to go back to the trigonometry textbook and from high school and look all this stuff up. Unfortunately, several of the pages are kind of crumbling now in that book. I might have to recharge. So the longer the overdominance ratio is, the length of the overdominance vector relative to the additive vector, the greater the region of morphological space for which the combination of traits is overdominant. The O vector will be longer when the D vector is longer, or it forms an angle with the A vector closer to 90 degrees. So the closer that gets to 90 degrees, or the longer the dominance vector gets. So here you can see a short versus a long dominance vector, and here you have a smaller angle and a larger angle. All that leads to more possibilities for overdominance. So if you look at this system of dominance here, with the different dominance for different traits, we can imagine what would happen under selection. So here's that same situation I've been drawing. And let's say we have selection, directional selection, for X to get bigger and Y to get smaller. Since the this is the usual situation for our QTLs, since most additive vectors are size related, being positive, positive, directional selection on shape factors might often lead to balancing selection on pleiotropic genes affecting the trait. So this represents a shape vector. This would be a size vector. There's the size vector. So we might expect selection on shape would result in balancing selection on loci affecting these traits that have differential dominance. The amount of morphological space in which directional phenotypic selection leads to stabilizing selection on the individual locus increases with the overdominance ratio. So when you have a small angle like this here in a short dominance vector, this space here, if the selection vector was within this region, you would have balancing selection on the locus. If it's outside of the blue region, it's not. See this, even though there is no overdominance at trait X or trait Y. Over here, we see a longer O vector and a bigger angle, so that a majority of the space in that direction can have overdominant selection. So we looked at our mandible traits, and we looked for this differential dominance. And we looked at the overdominance ratios for all the mandibular QTLs, and this is a frequency distribution of the overdominance ratio. And again, the 0 here is because of statistical significance. We're not able to detect the small effects with the sample. But the mean overdominance ratio is 0.71, right out here. And about 40% of directional selection vectors would produce overdominance for fitness at these QTL, on average. So that's a pretty substantial morphological space. 60% with this system, with our traits, no overdominance. 40% of the morphological space, you have overdominance to be selected on. This is all the mandibular QTLs that have significant dominance vectors. The other one was all the QTLs, whether they had any dominance or not. Now it's only if at least one of the traits has a dominance that's significant. And here the mean goes up to 0.89, and about 47% of the directional selection vectors will produce overdominance for fitness. This is to show you that what I've been talking about is not a strange idea at all. It's the way you get overdominance, is by having dominance relations different for different traits. So that's why the capital A and the small a can't represent dominance. You have to tell me what the trait is, because the dominance will change from one trait to the next. So this is the example of balancing selection in humans. Hemoglobin locus and the sickle cell anemia allele, where you have selection for malarial resistance and selection against anemia. So we would have that downward facing selection gradient. And if we look here at the genotypes and the phenotypes, we would have anemia. And for anemia, capital A is dominant to S. Right? The only individuals who get anemia are really the sickle cell homozygote. Malarial resistant, S is dominant to A, because these two genotypes are resistant to malaria, and this one isn't. If we then look at the fitness of these genotypes, you find that the fitness of the heterozygote is higher than the fitness of either homozygote. And that leads to balancing selection and the maintenance of an intermediate allele frequency for A and S in the population. Well, this is the phenotypic results here drawn out. Anemia, malarial resistance, two different traits. We plot the capital A homozygote, the capital S homozygote. And then there's an O vector here. And fitness lies along this line. So there's overdominance for fitness because the selection is for resistance and against anemia. So the same system I'm describing for mandibular traits also, it's the common situation. You think of balancing selection, that's what's going on. And this is just a different way of representing it. So it looks like balancing selection could be quite common in nature, especially for our morphological QTLs if that selection is on the shape of the organ. All we have to do would be to find those individual genes that have this differential dominance, and that's what we did when we mapped. We found that there was a lot of variation in dominance across the traits. There's the selection I forgot to press the button. So that was epistasis and dominance with pleiotropy. I'm sorry, that was dominance and pleiotropy. We show how when you have both variation in dominance and pleiotropy, you can get a situation where the heterozygote can have an overdominance for fitness, and variation at those loci could be maintained by directional selection on the phenotype, resulting in balancing selection on the genotype. So that's not stabilizing selection that's going on there, it's all directional selection. So I'm also going to talk about what happens when you have epistasis and pleiotropy. And again, we're going to talk about a situation where you have pleiotropy, you have genes affecting multiple traits, and you have epistasis that is different from trait to trait. Just like we had dominance differ from trait to trait, now we're going to have epistasis differ from trait to trait at a pair of loci. So you may have an epistatic interaction for one trait and not for the other. Or you may have epistatic interactions for both traits, but one might be additive by additive and the other dominance by dominance. They would be a different pattern of epistasis. So pleiotropy can be genetically variable when there's differential epistasis at pairs of loci. Differential epistasis occurs when the epistatic effects of a pair of loci vary across a series of traits. With differential epistasis, the pleiotropic effects of a locus are genetically variable, and since they're genetically variable, they can evolve. So again, this is going to be an example with natural selection, as the differential dominance was an example relating to natural selection. And here we can see the role that epistasis plays in terms of natural selection on pleiotropy. And basically, epistasis causes variation in gene effects at a locus. And those effects can be for a single trait, or they could be for multiple traits. This isn't really like a brand new idea. It's been around a lot. We're just going to deal with a different formulation of it. This is an example of correlation coefficients between cholesterol and triglycerides here. Correlation between cholesterol levels and triglyceride levels. Now, in the human population, these are usually considered to be reasonably highly correlated phenotypes. If you have high cholesterol, you're also likely to have high triglyceride. But if you look at the ApoE genotypes, alleles 2, 3, and 4 are the ones referred to, you can see that the correlation varies dramatically depending on the genotype. and that when you're carrying a 4 allele, there's no correlation at all. So here, the correlation coefficient between two phenotypes changes depending on the genotype at the locus. Uncorrelated if you're a 3-4 or 4-4, moderately to highly correlated if you're a 2-4, 3-3, and 2-3. 2-2s are relatively rare in this population, so that's why they're not on the chart. So the relationship between cholesterol and triglycerides, whether they go up together or not, depends on the ApoE genotype. This is an example from Alan Templeton's work on abnormal abdomen. He was studying Drosophila in Hawaii, not Hawaiian Drosophila, but cactophilic Drosophila from Central America, I think, that made their way to Hawaii along with the cattle populations and the cactus. And he studies them under a gradient of environments. So there are cactus that are at the bottom of a hill where it's really dry and hot, and at the top of the hill where it's very moist. The trick was to look at this from the top of the hill. You went to the bottom of the hill and had to walk back up. It was terrible. Hard work. You think Hawaii, it's nice. Instead they're crawling around in chaps amongst the cacti, and the grade is like 45% on the hill. So, in the natural population, they had this abnormal abdomen mutation, which is, I think it's our ribosomal DNA repeats, how many repeats you have. And in the laboratory populations, this mutation was discovered because it produces an abnormal abdomen cuticle. So the cuticle, the abdominal cuticle never hardens. It stays soft. They have a slow development time, high early fecundity, and low adult survivorship. So he's looking at the natural population, and they found the same kind of allele in the natural population. But it didn't cause abnormal abdomen, even though molecularly it was the same allele. Instead, it had a normal cuticle, slow development time, high early fecundity, and low adult survivorship. This was the set of features that you would want for life history traits at, I think it was at the bottom of the hill, where it was dry. If you didn't have a normal cuticle, if you had a soft cuticle, you wouldn't survive long in the dry, hot environment. What he discovered is that there were modifier genes in the natural population that suppressed the expression of juvenilized cuticle without modifying the life history effects. So the pleiotropic range of abnormal abdomen, you might consider this its normal range, that's how it was discovered and named, is restricted, so it no longer affects the cuticle it's named for. It still affects life history, but not the cuticle. So the effect of abnormal abdomen is evolved in this natural population to lose its cuticular effect. Now here's an example out of agricultural genetics that I steal from Brian Logden's. And these are corn kernels. And they were interested in trying to have a high lysine content in the corn because it's an essential amino acid. And so you got to eat it, you can't make it. So they had the wild type corn here. And then they had an opaque 2 mutant. And this mutant had very high lysine content. But you can also see that it has a very thin endosperm. It's a soft, so this is soft while this would be hard. You put this in the corn crib and it would all turn to mush. So it's not harvestable, whereas this is. So what they wanted to do was get the high lysine with a thicker coat. So they selected amongst opaque 2 mutants for individuals that had thicker coats. And that selection produced evolution at modifier loci that modify the opaque 2 mutants and produce a high lysine in the thick coat. So here they, by artificial selection, they limited the pleiotropic range of traits affected by the opaque 2 mutants. So it retained the good effect and eliminated the bad effect. This is just another example where we have the 2 traits X and Y. We have a target locus genotype, goes from small a to capital A. And we have a modifier locus that's capital M in this population and small m in that population. So in this population, the A locus has an effect on both traits because the capital M is present. The small m is present. You only have an effect of the A locus on trait X. It's not affecting trait Y. Now let's not look at that. Isn't that beautiful? I'll go to some real examples. Oh, okay. I'll go to some real examples instead. For time's sake. This again is a beautiful equation for estimating what we call relationship QTLs. So regular old QTLs affect the mean. The genotypes affect the mean of the trait. It's pretty simple. These relationship QTLs may not affect the mean at all, but they do affect the relationships between traits. The correlation between traits changes according to the genotype. And this mapping equation basically shows how you would do that. We used mandible length and then looked at the correlations of all the submandibular measurements that I showed at the beginning, and looked at all those in relation to mandible length. So if the model is significant, you can say that the QTL affects the allometric slope and correlation of specific mandibular traits in relation to overall mandible length. So just like we found 26 QTLs before when we were doing the morphology of the mandible, here we got 23 QTLs that show changes in the correlation between traits. They're on 13 chromosomes. 30% of these 23 are in the same location as the QTL that affects the mean. These RQTL traits affect, or loci, affect 1 to 10 traits each. 8 QTLs affect 1 trait, while the remaining 15 affect an average of 4 traits each. All the traits are affected, with some traits being very rarely affected, only twice, and others being mandibular height and superior incisor alveolar length, having many interactions. Traits that are affected by the individual relationship QTLs, when it has multiple traits, tend to be restricted to developmentally distinct mandibular regions, either the muscular process or the alveolus. So just like we found modular effects on the mandible, here there are also modular effects of these relationship QTLs, gene locations that affect the relationship between the individual traits and mandible length. So these are the 2 measurements that showed a lot of interactions, 3 measurements that showed a lot of interactions with the mandible length. So this is a plot, again, of the regular QTLs in blue and of the RQTLs in red. And you can see that in most instances it's a different location. This is to give you an idea of what I'm talking about with a real example. We have mandibular height and mandibular length. And we regress mandibular height onto mandibular length. In each of the 3 genotypes, we do it separately, in each of the 3 genotypes, at a location where the interaction has told us that the relationship between mandible length and height interact with the genotype. So here we have the large genotype, the small, the dotted line, and the heterozygote shows an intermediate relationship. These are the slopes of these lines. The same relationship would hold for the correlations. We have a basically uncorrelated S genotype, homozygote, and a more highly correlated L genotype. You'll notice that these lines cross. This is indicative of the interaction. The interaction here is between the genotype and mandible length. If you have a short mandible, the large allele produces a low height, low mandibular height. The small allele gives you a very high mandible for that same length. However, if you have a long mandible, the effects of the alleles are switched so that the small allele makes for a lower mandibular height and the large allele for a higher mandibular height. So does this locus, D17, MIT16, does it affect the posterior corpus height? It didn't show up in the genome scan. But it actually does. It's just that it's the negative effects of the L allele for small mandibles are counterbalanced by the positive effects of large mandibles. And they cancel each other out and you see no effect. Because you're looking at the general scan here at the average mandible length. And at the average mandible length, they're all right on top of each other. Does that make sense? It's important that the crossing occurs within the range of mandible lengths in the population. So that none of these are extended out to infinity or anything. The X and Y axis represent the range of variation in the population. And they do cross within the range of variation in the population. Here's a second one where we have the inferior condylar length and mandible length. And again, we see basically the same pattern we saw before. The large has a higher slope and correlation than the small, the heterozygote intermediate. And here we have a little bit different one. This one has the heterozygote has the steep slope and the strong correlation, while the homozygotes do not. And here we have the opposite of the pattern we first saw, where the large allele makes you have a longer anterior inferior basal length. There, makes that longer if you're in a short mandible than if you're in a long mandible. Here, this locus is detected on the genome scan originally, because at the mean, you can see that the genotypes are differentiated at the mean mandible length. So we did pick up this in the main scan as affecting the anterior inferior basal length. So this kinds of studies have been done on lots of different traits in our lab. Well, we found with the mandible, Mihaela Pavlichev has been looking at the long bones, where here she has the length of the humerus, tibia, and there's also examples from the femur and the ulna. And here we can see these differences in slope again when in relation to body size. That the allometry of length of the tibia versus body size changes depending on the genotype at this locus. So these two are the same locus. And here, at least the tibia and humerus both show the same pattern. This is an example to show you what the graphs showing you look like, along with an epistasis plot. So this relationship QTL is caused by epistatic interactions with the genetic background in general. And that epistatic interaction with the background has to be different for the two different traits in order to get this relationship QTL to come about. So this is showing how across the three genotypes at this other locus, you have different slopes for the genotypic values, and how those then project onto the XY plane. So that it's different epistasis for trait X and trait Y that result in the RQTL. Now here's a bunch more interesting examples. These differences in slope and correlation have two different causes. One is you could have a change in the variance of the size trait here, the body size trait. If the genotypes vary in the variance of the body size trait, that's going to cause a difference in the slope and in the correlation, because the slope has the variance of body size in the denominator. And it can also occur due to changes in the covariance between the two traits, in which case the variance of body size might remain the same, and the covariance be responsible for the changes in relationship. So that's what's kind of shown here, where you can see some changes in the variance of body size and how that sometimes produces the RQTL relations versus other cases where it's actually more the covariance that's changing. Both will change in these situations. It's not an either-or situation. This is where they lie, and then where the epistatic QTLs lie along the genome as well. So the red ones are RQTLs for long bone length with body size, and the green ones are their epistatic interacting loci that have different epistatic patterns for the different traits. So we're going to go a step back here to that very first example I showed, where we had cholesterol and triglyceride levels, the well-known positive correlation between them, really only being true for the most frequent genotypes in the population. But some of the less frequent genotypes don't have any association between triglyceride level and cholesterol level. Now here instead, this is going to be cholesterol level or triglyceride level in relation by ApoE genotype again. But here, here is the exact same thing done in different European American and African American populations. And you see the same result. If you have a 4 genotype, your it's a little confusing there, but the 0 correlation is here. I don't know why my computer did that. It's probably some Macintosh invasion of my Windows computer that did that. But you can see the same changes between genotypes in correlation that we saw in that very first slide. But here he's got more genotypes present, got all of them, because the sample size is much bigger. So this replicates that first result. The correlation between triglyceride and cholesterol levels is affected by the ApoE locus genotypes. This shows triglyceride and total cholesterol again. But instead of being a correlation in relation to each other, here's the correlation between triglyceride level and coronary heart disease. And likewise between cholesterol level and heart disease. And what you can see in the graph here, this is actually done with a survivorship curve, an exponential hazard coefficient. A coefficient of 1 indicates no risk. Coefficients above 1 indicate high risk. Below 1 indicate kind of protective level. And so it's like a correlation, but it's a different parameter. And you can see here that some of the genotypes don't show a correlation, or much of a correlation of risk between the level of triglycerides and cholesterol and risk of heart disease. so that the risk of heart disease relationship with cholesterol and triglyceride that we treat as known and common for everybody is conditional on the ApoE locus genotypes. And again, it's if you have a 4 in your genotype, you tend to have no risk associated with high cholesterol or high triglyceride. If anything, you got a bit of protection against heart disease. But these aren't significantly lower than the line of 1, so that would be over-interpreting things. So you got a group of people here who perhaps lowering their triglyceride and cholesterol levels by medication is not going to make any difference in their coronary heart disease risk. So you might lower their cholesterol, might lower their triglyceride, but in terms of heart disease, you're not doing them any favors. Whereas there are other people with a clear and strong relationship between heart disease and cholesterol levels and triglyceride levels. So that's supposed to be individual medicine. That makes all of the mandible QTLs like relevant to the real world. So gene effects on specific mandibular traits or long bone lengths and so on are often context dependent, and that they vary depending on the length of the mandible or the size of the body or some other phenotypic character. The range of traits affected by a pleiotropic locus is subject to genetic variation. So whether a locus affects one trait or not is going to depend on epistatic interactions with other loci. Other loci can cause additional traits to be linked to your locus, or it can take away traits linked to your locus. And genetic variation in pleiotropy is due to this differential epistasis. So, a hypothesis that we had is that modular pleiotropic patterns, which we saw was the case here, are sculpted by directional selection so that co-selected traits come under the control of a common set of module-specific genes, while traits under independent selection come under the control of separate sets of genes. So that again we have selection modules determining the pleiotropy modules, which then in turn determines co-inheritance and co-evolution of traits. So this is my version of the model. I was visiting Jason in England, and I went to a wonderful concert, Brown Eyed Girl, fantastic. And not being English, the ticket said the concert would be at 8. So I showed up at 7:30 to get there early, and 8 o'clock is when the doors open. The ticket that time is not when the concert starts, it's when the doors open. So I had extra time to kill. Fortunately, there was a bar, and there was gin and tonic available. So I took advantage of the bar, and then I worked out the theory right here on the envelope. There's another theory on the other side of the envelope, but it's no good. So this is my version of the model, and it's a great version. The idea here would be that these RQTLs show differences in correlations between genotypes. So here we have, if you're capital A, you don't have any correlation. If you're small a, you got a pretty strong correlation. And no correlation, strong correlation. And we're going to look at selection on these, directional selection on these traits, X and Y. And here you can see we have 3 genotypes with their distribution superimposed. And what's going to be important here is the direction of selection relative to the major axis of the distribution of the traits. So you see the major axis of this genotype runs this way. So does the heterozygote. The other homozygote is a circle. So it doesn't have any correlation. It doesn't have a major axis really, so it has no alignment along any direction. And this is a plot of these genotypic effects in cross-section at the X minus Y trait, which is representing selection here, X minus Y. X to be small and Y to be big in this case. And here you see that we have along the selection vector, but there's different levels of variance for the three genotypes. Capital A is a wide variance, that's the zero correlation. Heterozygote and intermediate variance. And the small a homozygote has a small variance. The variance being how long the axis is under the directional selection factor. If you then impose a nonlinear selection on this system, and everybody above the threshold mates and has kids, and everybody below the threshold doesn't. This is a typical thing used in agriculture all the time. I don't know if you can see it very well. I should have made the lines thicker, I guess. But you can see that a larger proportion of the AA genotype is to the right of the threshold than of the small a genotype, where only a small portion are beyond the threshold. So the selected population will be enriched for the capital A allele. And the selected population will show a higher correlation between, or a lower correlation between X and Y than you get with the small a, and heterozygote. And it doesn't really matter how your nonlinearity is specified, it will be true as long as it's nonlinear. So here we have our three genotypes: no correlation, moderate correlation, high correlation. And we're going to select for a lower correlation, essentially. We're going to select along that X minus Y or Y minus X axis. So after selection, after the first generation or two, we have an enhancement of the no correlation group and a diminishing of the heterozygote and high correlation group until eventually everybody is now fixed at the RQTL for the no correlation genotype. So in this population, there would no longer be a correlation between X and Y. Whichever genotype has the best alignment with the direction of selection, that will produce a change in the variation pattern so that it aligns with selection and increases the variance in the direction of selection. The epistatic interactions are changing genotypes. And through that method, it's increasing the variation along the direction of selection. So you're producing new adaptive variation through this epistatic mechanism. This shows, again, this is the situation we just talked about, the Y minus X. And these are the frequencies of the no correlation and the high correlation genotype, and this is the level of correlation. You can see in this system, you would be selecting the correlation down to 0, and the 0 class genotype, the capital A, would fix, while the other genotype will be driven to extinction. If instead we have selection aligning along the major axis of the small a, the opposite would occur. Then the small a genotype that lines up nicely, it would be favored, and it would go to fixation. And the population would end up with a high correlation between X and Y, two co-selected traits. This should be Y plus X. That's another one of those minus plus sign mutations. Now, you saw my model. That's my model. And it's a beautiful model. It has no basis in math. It's all intuitive. I took a couple of years to talk Gunther Wagner and Mihailo Pavlichev into doing a formal mathematical model of the process. And that came out last year in the Royal Society. Now, Gunther explained that while I don't really care about haploids, that the haploid model would be a lot easier than the diploid model. So it's a haploid model. There are two genotypes, locus A with allele I and allele J, and they have a different level of correlation. And here we have trait Z1 and trait Z2. So we're looking at a situation with an RQTL effect on variance and covariance. You see this difference between the red ellipse and the green circle, but no effect on the mean of the population at all. The two distributions are right on top of each other when it comes to the mean, where the star is. And that it's a haploid organism. This shows a three-dimensional plot of the same thing with fitness added in. So we have the first trait and the second trait. Here's our two ellipses for the two traits. We're looking at it at a bit of an angle, so that's actually round if you looked at it from above instead of from the side. And here we have the log of fitness. And we're going to say that the fitness increases as Z1 increases and as Z2 increases. Fitness increases. Wow. These are Z's, representing the trait, regardless of the fact that there doesn't appear to be any of the middle part of the Z. That's what they are in the computer. And these are the genetic variance covariance matrices for each allele. And under weak fitness, we're going to just say that the average fitness of the two alleles under weak selection is going to be the same, and that's got to do with the no difference in means. If there are differences in means, then that alone would cause a change. And this shows in later generations, and the point of showing this is that change in fitness, which is going to represent change in mean fitness, the evolution, is going to be affected by the strength of directional selection in direction 1 times the difference in genotypic variances for allele I and J, 2 times the strength of selection in direction 1 times the strength of selection in direction 2 times the difference in genetic covariance. Again, this should be, well, this says J and J, but it shouldn't say that. It should say I and J. And again here, selection in the second direction and the difference in variance for the I and the J here. This would be the difference in covariance between the two. All of that is put together to determine the evolutionary change, along with a term that's L. And this L term, turns out, was not in my intuitive mind, even though it seemed to be intuitive to Gunther Wagner that it was in the model. It wasn't to me. L is the level of linkage disequilibrium between the RQTL and other loci that affect the mean of the trait. A value of 0.75 is no linkage disequilibrium. A value of 1 is for full linkage disequilibrium. So this L equals 1 is going to be the situation where a trait affects both the mean and the variance covariance. The genotypes both mean and variance covariance differently. And in this selection regime, the selective advantage of an RQTL depends on the level of its linkage disequilibrium between the RQTL and the other loci affecting the mean. So it will be the differences in relation to directional selection here, along with the average linkage disequilibrium with the genetic background. And just like before, though, when you try a positive selection, you have correlated and uncorrelated here, you're going to get the evolution of a stronger correlation. The gene frequency will fix for the genotype that has the correlation consistent with the directional selection. You can see that the rate of that evolution is affected by the level of linkage disequilibrium. 0.75 being lots of linkage disequilibrium and 1 being very little linkage disequilibrium or none. I cannot explain to you why 0.75 is a lot and 1 is nothing. You must talk to Gunther if you want to know about that. He explained it to me several times. So if we think of it in a more dynamic evolutionary sense, we start out here with the two genotypes. And then over the generations, the correlation will change so that the major axis of variation lies along the direction of selection, leading to an increased rate of evolution. So here, traits that are selected together are going to come under the control of common pleiotropic loci. And since they're under the control of common pleiotropic loci, they're going to be inherited together, they'll be heritable together, and they'll also evolve together. The same process works in reverse. If you have a high correlation here, and you're selecting for opposite directions, you're going to drive the correlation between the traits down, and even negative if such variation exists in the pleiotropic effects of the alleles. So you can both build and destroy modules by this mechanism. And it's using variation that we see as quite common around our mouse world anyway. It's pretty common. And you even see it with these strange human genetics results too. So if such variation is common, what we would see is that natural selection's directional selection would sculpt the pleiotropic patterns to match function, and thereby arrange so that the genetic variance covariance matrix is a representation of the functional interaction matrix, or developmental interaction. Well, here's more. I think there's more. This actually indicates you don't just increase variance in the direction of selection, you also increase it in the opposite direction. It's along that axis. It's not all at one end of the axis, because there's no change in the mean. So this is a situation where the directional selection switches from positive to negative through time. And you can kind of see that here, positive and negative through time. And what that will do is the same thing the directional selection did. It's fluctuating directional selection. As long as it's along the same axis, but going positively and negatively, will also induce correlation between co-selected traits. So even if over the long term there's stasis and stabilizing selection, each generation at a different temperature, a different water level, could lead to selection positively and negatively depending on the environment. And that would also, though, co-selected traits that function together are going to become correlated through this mechanism. That wasn't in line either. So RQTL selection can explain patterns of correlation. Co-selection on traits leads to positive correlation. Antagonistic selection leads to negative correlation or null correlation. Selection on only one character and not the other selects for a decoupling of the traits. So if originally there are pleiotropic effects spread across all the traits, but now I'm just going to select on this one, it will end up making that trait independent of all the others. The pleiotropic effects will be diminished so that it only affects that one trait. And this RQTL selection works under sustained directional selection and under fluctuating selection. And there's a picture of Mihaela, and there's a picture of Gunter, and there's other stuff. And now I know you guys want to look at this again, because this I guess there wasn't complete brown in my beer, but there's more than there is now. I already had these. All right, so if there are any questions, we'll take them, and that should be it for now.